title slide, okay, everybody? Okay, wonderful. So yes, thank you for introduction. And this is the title of my talk that was already introduced. This is just a disclaimer that any views that I'm presenting are solely those of mine, and PerkinElmer Genomics is not responsible for accuracy and reliability. So more than 7,000 known diseases are very rare. Majority of them are caused by pathogenic mutations in one of our 20,000 genes, each affecting only a handful of people across the world. While individually scarce, collectively, those disorders constitute significant burden. So by the recent estimates, more than 10% of US population are affected by rare disease at some point during their lifetime. Rare diseases tend to be chronic, and they also can be detrimental. They underlie more than a third of deaths during infancy. So collectively, they are also costly. It is much more expensive to treat a person with a rare disease, and they also as a group constitute more than 10% of inpatient costs. Now, because those disorders are so rare and highly diverse, there is in general a lack of awareness and knowledge among physicians, which contributes to a diagnostic odyssey that patients usually have to go through. Based on recent estimates, on average, a patient visits around 17 specialists, and it may take 7 to 10 years to get a diagnosis. And this all adds up to adverse outcomes. Up to 20% of patients with rare diseases, they suffer from missed, incorrect, or delayed diagnosis. And this may mean that the disease they have may progress beyond the point of successful treatment. They may receive inappropriate management, and you can only think about the emotional toll that the patient and their caregivers are going through. And it's always good to put some numbers to visualize even further. And here are the numbers of economic burden of rare diseases. In US alone, in 2019, rare diseases costs reached $1 trillion. Now, 400 billion went to direct medical costs, such as inpatient and outpatient services. But more significantly, indirect costs that are associated primarily with loss of productivity and other non-medical categories racked up to more than half of this expense. So bottom line, rare diseases as a group come with significant burden, not only to the individuals that have them, but also to the society as a whole. And therefore, we should continue working on diagnostic and treatment solutions to alleviate this burden. And so now I work at the PerkinElmer Genomics, and PerkinElmer Genomics specializes in genomic diagnostics of inherited disorders and therefore rare disorders. We as a company grew out of the newborn screening lab with rather limited diagnostic services for newborns only into a lab that currently offers multifaceted clinical solutions throughout the lifetime of an individual. And so this includes prenatal and postnatal testing, as well as diagnostic services for families, including adults, who are often disregarded or forgotten in this field. So in the lab, we use a plethora of technology platforms to enhance our diagnostic capabilities, and this includes DNA sequencing, biochemistry assays, as well as others. However, in this talk, I'm only going to talk about whole genome sequencing and to show how it can provide medically relevant information throughout the continuum of care, from prenatal to adult stages. So what is our power at PerkinElmer Genomics? Well, efficiency and quality. We have unsurpassed capacity for sequencing with no downtime. And we also have automated solutions for every step of the NGS wet bench workflow. Now, the efficiency and quality transcends into our dry lab operations that carries out data analysis, interpretation, and reporting. We have internal bioinformatics solutions to enhance our dry lab production efforts, and we have quality personnel. Sequence variants, interpretations are performed by highly skilled PhDs, and every single test is reviewed and reported out by board-certified clinical genomic experts. Now, we're also highly flexible. And this applies for the sample types that we accept for sequencing, with dried blood spots being our staple. Now, dried blood spots is the sample of choice for the neonatal population, as every newborn, as you know, gets sampled for mandatory newborn screening purposes, at least in the US and Europe, I guess. And the dried blood spots are also easy to transport. Now, it is worth mentioning that we, to my knowledge, are the only commercial lab that is New York State approved to perform NGS on dried blood spots. So we also are highly flexible in NGS testing options that we offer. We offer various size curated panels, as well as we take orders for custom panels. We also do whole exome sequencing, whole genome sequencing tests, and we also participate in sponsored testing programs, such as neuromuscular testing via Lantern project or Anemia AD, and so on. Last but not least, we are a global lab. Our laboratory headquarters are in Pittsburgh, but we have labs across the world, including China, India, Malaysia, and Sweden. Now, the biggest, but most likely not the most obvious advantage of this is that we have access to genome data from diverse human populations. And this is extremely important. As you know, different populations differ in their genomic makeup, which has to be taken into consideration when providing clinically relevant interpretation of the genomic findings from the testing. And so majority of our whole genome sequencing, and I'm going to abbreviate this as WGS, is done for diagnostic purposes. And that is for clinically affected children and adults. So we offer two options. One is CN-genome, which is a low-pass genome sequence that can be considered as a replacement for microarrays, but just with a better resolution. So this test is used for copy number variant calling, has a capacity to pick up certain chromosomal rearrangements, as well as uniparental dice. Now, our clinical whole genome sequencing test is a phenotype-genotype-driven testing. So whole genome sequencing results in a relatively uniform, or we produce a whole genome sequence of a relatively uniform coverage of 40x on average for the whole nuclear genome, which enables us to call sequence and copy number variants in both coding and non-coding sequences. Now, in addition, our test includes mitochondrial DNA sequencing that results in more than 1000x coverage. And this enables a reliable detection of variants at a very low heteroplasmy levels, as low as 5%. In addition to this, our test includes bioinformatic solutions to screen genes associated with trinucleotide repeat expansion, as well as to screen SMN1 gene, which is associated with spinal muscular atrophy, for a copy number. Now, I would like to know that these two options are not diagnostic, and they require post-WGS confirmatory testing. In addition to receiving diagnostic findings that are relevant for clinical indication for a patient, the patients can also opt in to get a clinical significant variance on medically actionable genes as recommended by American College of Medical Genetics. They can get also reports on incidental diagnosis if found, as well as pharmacogenomic variants, and reports on their carrier status in over 300 genes. In addition to the clinical genome for people with diseases, we also offer whole genome sequencing for pediatric healthy screening purposes. And this is done via ViaCord, can be ordered via ViaCord, our partner. And we also offer WGS for both healthy adults and children screening on research basis in partnership with other institutions. So the latest addition, which we are very proud of, to our WGS menu is ultra-rapid whole genome sequencing. And as you know, while speed comes with increased price, the clinical utility of urgent WGS have been well documented for critically ill babies in NICU and PICU by 2 pilot studies. So they showed that whole genome sequencing test yielded diagnosis for 40% of those critically ill babies, with more than a third of the babies receiving changing care. So answers were giving faster, the stay in the hospital shorter, the medical management more appropriate, and all of it came down to significant savings in healthcare costs. Now, our ultra-rapid whole genome sequencing includes a phenotype-genotype-driven analysis of nuclear and mitochondrial genome, and it also includes a comprehensive biochemical panel of more than 70 diseases. And all of this it has a turnaround time of less than 5 days, which is really fast. So this is just a very quick look at our preliminary diagnostic landscape of the initial set of clinical whole genome cases. So the work on a much larger data set that we've accumulated over years is in progress, and we intend to present it in the upcoming American Society of Human Genetics. Now, as you can see, the significant part of our whole genome testing is done for adults. So here they constitute almost a quarter of the cases. Now, potential diagnoses were identified across the whole cohort in over 40% of patients. And this is just what type of variants were found. Like, no surprise, the sequence variants constituted majority of diagnoses, so roughly like 80% and more, while CN copy number variants only constituted 10% of diagnosis, and roughly around 2% of individuals received dual diagnosis. So that means that pathogenic variants in 2 genes seem to be responsible for the phenotypic clinical indications that they came from. And it's always I'd like to present some case studies that just visualize or demonstrate the power and the impact of the whole genome sequencing. So the first case is our ultra-rapid genome testing. So this was done for a 2-day-old male. The baby had a multiple congenital anomalies. So had hypotonia, the abnormal MRI findings, breathing difficulties, metabolic abnormalities, some skeletal findings, as well as non-specific dysmorphism. Now, our ultra-rapid whole genome sequencing identified a hemizygous missense in PDH1 gene that was classified as likely pathogenic. Now, this finding, without going into too many details, is consistent with diagnosis of pyruvate dehydrogenase complex deficiency. And this leads to a mitochondrial defect impacting the ability to metabolize sugars. And as you can guess, it is not good. And this results in energy production defects that have multi-organ implications. Now, due to our super fast whole genome testing, which was completed in this case within 53 hours and during the weekend, it's not that we prefer it, this patient is in the best position as he can be. Already at the day 5 of his life, he can receive appropriate medical care, and the family has the answer, which often comes with a big relief. Now, the second study was done for a 20-year-old South Asian male. He had distal muscle weakness, muscle atrophy, gait difficulties, and the clinicians highly suspected a Nonaka myopathy with the disease that had a certain characteristic features. So the sequence analysis of the whole genome sequencing for this individual identified a heterozygous pathogenic missense in a GNE gene, which is responsible for autosomal recessive Nonaka myopathy. But this is just one mutation, and it's autosomal recessive, so we would need 2 to explain the phenotype. So the copy number variant analysis for this individual showed that this patient had a 11.5 kilobase deletion, intronic deletion in this gene. Now, I'm just showing a diagram of GNE gene. exons, the boxes are exons 1, 2, and 3 going the other way than we usually used to. And this shows a coverage of NGS reads that are mapped to the reference sequence. So as you can see, the coverage drops at this position, indicating that there are fewer reads that map to the reference, were generated that map to this reference region, and therefore that indicates a deletion. So this deletion is smack in the middle of intron 1 of this gene. Now, digging through our internal database, we identified the same deletion in 2 other South Asian patients with clinical indication for myopathy. Also, a similar size deletion in the intron was reported in scientific literature and was shown to affect GNE expression in muscle biopsy of the affected individuals. And this all evidence information served as a sufficient evidence to classify this deletion as likely pathogenic, which ended the diagnostic odyssey for this 20-year-old male. And finally, the last case I'd like to present is for a 15-year-old proband. This was a testing for a family trio, meaning a mother and father samples were also submitted. So the family requested or opted in for all optional findings. And so the search for other diagnoses revealed a maternally inherited pathogenic loss of function variant. You can see it's a deletion of a 1 nucleotide, which would lead to a out-of-frame transcript in a parking gene, which is associated with autosomal recessive juvenile type Parkinson disease. But hey, this is just one hit, and many of us have a 1 pathogenic allele in a gene that is responsible for autosomal recessive condition, but that does not make us sick, and that just makes us a carrier. Is this the case for this patient? Well, our further analysis of and in this case, CNV analysis, revealed a paternally inherited duplication of exon 2 in parking gene. So here's again a diagram of a parking gene going the other way, 1, 2, 3, and so on. These are the coverage of NGS reads that as they are mapped to the reference sequence. So you see that there are more reads, suddenly a bump of read coverage that indicates that this genomic segment in this individual is present in more copies than in the reference sequence, indicating a gain of this material. However, this does not tell us anything about the actual position or orientation of this duplication. It could be anywhere inserted in a so-called junk genome region and would have nothing to do with any disease. And usually this type of uncertainty associated with duplications renders them classified as variants of unknown significance. And it's worth noting that if this was a whole exome sequencing, this duplication most likely would not have been picked up because this is just in 1 exon. And if it was picked up, most likely it would have been called a variant of unknown significance because of the mapping uncertainty and would not have been reported in the settings of secondary or incidental diagnosis. But in our case, we have whole genome sequencing. We did whole genome sequencing, and that means that we should have reads that cover the break point in this individual. So here is again, I'm just showing a view or how we visualize our NGS reads, and these are reads that are mapped to the reference sequence. So the pink and the blue colors, they indicate that those reads have 100% match to the reference sequence. And what you can see, there are a few reads that I'm showing here. Half of them have a perfect match to a reference sequence, when the rest of the sequence, it's highly colorful, indicating that it does not match the reference, kind of the sequence that it's adjacent to it. So the thorough analysis of those reads helped us to actually map the breakpoints of a duplicated segment and which turned out to be inserted in tandem and in the same orientation as the native exon 2 in parking gene. So why is this important? And here it is why for those who like science. So the normal parking gene produces normal parking mRNA, produces normal parking protein. Now, intragenic tandem duplication of exon 2 creates parking gene that looks something like this. Now, exon 2, the duplicated exon 2, is likely to act just like a native exon 2 and would get incorporated into a parking messenger RNA. Unfortunately, this would lead to an out-of-frame product, out-of-frame transcript, which would result in the absence of parking protein. And therefore, ability to figure out the exact position of the duplicated segment helped us to move this duplication from the land of variant of unknown significance to a likely pathogenic variant. And now, what does it mean for this patient? Well, the patient has both, unfortunately, parking alleles defective, which leads which led to a report with incidental diagnosis of early onset Parkinson's disease. It's obviously a very grim news for the patient and the family, but with a silver lining, because as we all know, knowledge is a power. And so our whole genome test hopefully empowered the patient and their healthcare team to be well informed and prepared for the looming disease, rather than being ambushed by it somewhere later unexpectedly in life. So this is all that I wanted to show. And if there are any questions, I'm happy to answer them. Otherwise, this is my email that one can contact, and I would be happy to answer questions this way.
Thank you so much, Jorune.